Lights, Camera, Action Potential by Julia and Greg. Here is a photo of a neuron. Notable parts include the dendrites, where they receive the electrical message from the presynaptic terminal, the soma or cell body, where the message is continued, the axon hillock, where it generates a nerve impulse, the axon, where the myelin sheath that surrounds it insulates and allows the signal to be carried to the axon terminal efficiently. The axon terminal, where vesicles are located and can release things like neurotransmitters to transmit a signal in order to convey information to another cell. The neurotransmitter travels through the synaptic cleft, binding to the postsynaptic membrane. Neurons are important for the overall communication between cells. They communicate via a mechanism called action potential. Action potential is the change in voltage across a neuron's membrane where it carries an electrical signal. This signal is what allows the cells to be able to communicate with each other. Action potential has three main stages: depolarization, repolarization, and hyperpolarization. In this video, we will specifically be examining and explaining how acetylcholine is released from the neuron, transmitting the action potential signal to the next neuron. This is a picture of the legend used in this example. On the cell membrane of a neuron, there are many different voltage-gated ion channels which allow for the movement of sodium, potassium, and calcium ions to move in between the cell. These become important during action potential. Starting off, the resting membrane voltage is a normal negative 70 millivolts. The neuron at rest has a higher concentration of sodium ions outside the cell compared to inside the cell. Potassium ions, on the other hand, have a higher concentration inside the cell compared to outside the cell. Next, a stimulus will begin to depolarize the membrane to the minimum threshold of activation. This is roughly negative 50 millivolts. During this step, the voltage-gated sodium channels are the first ion channels to open. When enough of the sodium channels are opened concurrently in the same area, there is a significant voltage change in the local membrane. This is because sodium ions enter rapidly into the cell because of the concentration gradient of sodium ions. This is seen to be about 30 millivolts. This step is the depolarization of action potential. Because of the sodium influx, voltage-gated potassium channels open. At the same time, these sodium channels are slowly closing. The potassium ions move from inside the neuron to outside of the cell because of the concentration gradient, then repolarizes the membrane to go back to its negative resting potential, which is normally negative 70 millivolts. Once the cell membrane potential is repolarized, then the potassium channels begin to close. In the presynaptic terminal, the voltage-gated calcium channels are found. These channels open during the depolarization of the neuron. With the voltage-gated calcium channels activated, this allows calcium to move into the cell. As a result, it triggers the release of the acetylcholine that has been stored in the vesicles. The vesicles bind to the presynaptic terminal membrane, pushing the acetylcholine into the synaptic cleft. As the cells begin to recover to the resting state, the calcium channels begin to close. The acetylcholine then binds to the acetylcholine-gated sodium channels, which allows for sodium to enter into the postsynaptic membrane. This starts the depolarization step of action potential again, therefore continuing the message onto the next neuron.